Hello friend, welcome you all in the IIT PAL session of biomolecules. This is fourth session of biomolecule. This chapter is related in class 11th NCERT biology book. I BP Shah, PGT Bio, Kendriya Vidyalaya, Kesapuram. Before going to start my actual lecture, I have asked some questions related to my previous session. Let us look these questions. First question was, how many types of amino acid are found to occur in proteins? My next question was, name any two aromatic amino acids. Question number 3, which type of bond are found in the proteins and polysaccharides? Fourth question was, what does tertiary structure of a protein indicate? And last question, why is chitin also called fungus cellulose? Now, I hope all you will be able to answer these questions, because it is related with my previous class. Now, let us see the question first. How many types of amino acids are found to occur in the protein? Protein, which is known as polymer of amino acids, no doubt, and various types of amino acids linked together to form protein with the help of peptide bond. Now, we have already discussed there are 20 different types of amino acids are there, that amino acid linked together with the help of peptide bond and help in the formation of protein. Then the protein form different type of structure like primary structure, secondary structure, tertiary structure and quaternary structure. Okay. My second question was, name any two aromatic amino acids. Aromatic, as the term indicate, which gives smell. The two types of aromatic amino acids are phenylalanine and tyrosine. Let us see the third question, which type of bond are found in the protein and polysaccharide. We have already discussed in carbohydrate chapter. At the time of discussion in disaccharide like maltose, sucrose, lactose, these three disaccharides are formed with the help of two monosaccharide, and these two monosaccharide combine with the help of glycosidic bond. And in protein there are 20 amino acids, these 20 amino acids are linked together with the help of carboxyl group and amino group and form peptide bond. So, your answer is correct, that is, in protein peptide bonds are there and in polysaccharide there is glycosidic bond. Now, the third question, what does the tertiary structure of protein indicate? We have already seen in our last session different types of protein. One of the protein structure is tertiary structure of protein. This tertiary structure of protein indicate 3-dimensional globular arrangement of the polypeptide chain. Now, the last question, why is chitin also called fungal cellulose? Because in fungi its cell wall is made by chitin. So, because chitin form the cell wall of the most member of the fungi. So, these are the 5 questions which is related to the last topics. Now, I hope all questions which is related to these topics you will be able to answer this. Now, today's my session is lipid and nucleic acid. First of all, lipids. Lipids are known as heterogeneous group of compound which is formed just like carbohydrate, carbon, hydrogen and oxygen, where the oxygen is less in comparison to the carbohydrate. So, for the oxidation of lipid it requires more oxygen. So, lipids are known as heterogeneous compound which includes long chain of hydrocarbons. Lipid molecules hold large amount of energy and are energy storage molecules. So, lipids are also called energy storage molecules. Lipids are generally ester of fatty acids and are building block of biological membrane, because most of the membrane, plasma membranes or the cell membrane are made by lipids. Most of the lipids have polar and non-polar head and non-polar tail, just like this is the head region, this one is the tail region. This head region is known as polar head and this one is non-polar. Fatty acids just I have discussed in my last session. So, let us look. The fatty acid can be classified into unsaturated fatty acid and saturated fatty acid, because in saturated fatty acid there is single bond in between carbon and carbon, and in unsaturated in between carbon and carbon there is double and triple bonds are there. So, fatty acid has been classified into two main group, unsaturated and saturated. Bloor classified lipid based on their chemical composition, that are these are the classification. First of all, the lipids has been classified into simple lipids, conjugated lipids and derived lipids. Simple lipids, which is called homolipids, example fats and oils. It is also called triglycerides. These simple lipids are known as natural fat or oils, and second one part is waxes. Natural fats are made by fatty acid and glycerol. For the formation of one natural fat, that is simpler one is triglyceride, 3 molecules of fatty acid and 1 molecule of glycerol is required. If the glycerol as a alcohol, other alcohol combined with the fatty acid, then it forms waxes. So, waxes are for fatty acid and other alcohols. Now, the second one, conjugated fatty acid. These conjugated fatty acids are made by fatty acid, alcohol and other substance. This conjugated fatty acid has been defined into 3 broad categories, that is phospholipid, which contain other substance. Fatty acid and alcohol is very common, and in other substance there is the phosphate group. So, this group is called of conjugated lipid is called phospholipid, when phosphate group are present along with the fatty acid and alcohol. Next one is glycolipids, when the lipids, conjugated lipids are formed by fatty acid, alcohol and carbohydrate, that is called glycolipids. And third one is lipoprotein. These lipoprotein are made by fatty acid, alcohol and phospholipids and other proteins. So, the third one lipids are called derived lipids, which is formed by the hydrolysis of this one simple and the conjugated lipids. So, derived lipid can be defined as it is formed during the hydrolysis of simple and conjugated lipids. The example is sterols. We will look in this session. Now, the simply the third one derived lipids, its example is steroids and cholesterols. Now, glycolipids, first of all the glycolipids which included your syllabus. Glycolipids are the lipid whose head contain oligosaccharides with 1 to 15 saccharide residue. Phospholipids which contain a positively charged head which are linked to the negatively charged phosphates, and estrol whose head contain steroids ring, that is example is steroids. Now, first of all you will see the two main type of fatty acid that has been classified, that is saturated and unsaturated. The saturated fatty acid which I have already discussed, in between the carbon and carbon there is single bonds. So, this one is called saturated fatty acid. It has high melting point and boiling point. The example of saturated fatty acid you will write easily, that is CH3, CH2, 6 COOH. The first one fatty acid, that is crapolic acid, where the methyl group and CH2 group has 6 CH2, CH2, CH2 in the form of 6, then last one is COH group. If this CH2 group is multiplied by 8, then it form capric acid. If this CH2 group multiplied by 10, then it is called lauric acid. So, you see by multiplying the CH2 group 6, 8 and 10, we can easily form the different type of fatty acid that help in the formation of fat with the help of the bond that is called ester, which I have already discussed in my last session. The next one is, if CH3, CH2 multiplied by 12, that is called myristic acid. If that is multiplied by 14, that is very common palmitic acid, COH. So, palmitic acid have carbon 15, and if it is multiplied by 16, that form stearic acid. So, these are the some common saturated fatty acid. You can easily write down this chemical formula by knowing where the CH2 group multiply by either 6, 8, 10, 12, 14 or 16. The two common is very important for your syllabus, that is palmitic acid and the stearic acid. So, you can now easily write the structure of this saturated fatty acid. Now, look some unsaturated fatty acid. Unsaturated fatty acid. You can also easily write the structure of some unsaturated fatty acids are, you know, in between the carbon carbon bond there will be double and triple bonds are there. So, all the unsaturated fatty acid no doubt have double or triple bond in between that. Just CH2 when multiply by 7, here CH3, CH2 multiply by 3. This structure is called myrstolic acid. This is unsaturated because in between the carbon 1 3, at 5th carbon and 6th carbon there is a double bond. So, this is the structure of unsaturated fatty acid. If this one CH3 CH2 multiply by 5 and the same structure, here hydrogen is also present, CH CH2 7 COOH. Here in between the 7th and 8th carbon there is double bond. So, you can easily by replacing 3 by 5 and the same structure as the first one unsaturated fatty acid, this one structure is called palmiotolic acid. So, with the help of this structure you can easily write the saturated fatty acid and the unsaturated fatty acid by knowing where the double bond are present in which carbon atom and where it is multiplied by 7 or 5, 2, 4, 6, 8, 10, 12, 14 and 16. So, this is the structure of saturated fatty acid and unsaturated fatty acid. Now, you know the simply the fatty acid are formed by the carbon, hydrogen, oxygen, and this help in the formation of fat. How this fat is formed? The simple fatty acid which is made by 3 molecules of fatty acid when combined with glycerol, 1 molecule of 1 mole of glycerol, it form fat, that is the simple fat which is made by fatty acid and glycerol, that is called triglyceride. Now, have a look how this structure is formed. Chemically, the fat is defined as esters of glycerol and fatty acid or is a trisaccharides of fatty acids. Here the write the structure of glycerol. This glycerol contain 3 OH group, no doubt. We have seen the structure of glycerol in carbohydrate that contain 3 OH group, and here is the fatty acid, 3 molecules of fatty acid 1, 2 and 3. I have seen the structure of the palmitic acid which is made by C15 H31 COOH group. Now, from the glycerol and the acid group from the palmitic acid, it is going to be combined H and OH, H and OH, H and OH. Finally, 3 molecules of water is going to be released and the bond is formed. That bond, this one, this one, this one. This bond is called ester bond. You know, when the acid combined with the alcohol, that form ester. So, when the fatty acid combined with the glycerol molecule, 3 molecules of fatty acid, because the glycerol contain 3 OH group. So, it form the triglyceride, that is the simple structure, 3 water molecules is released and the fat is formed with the help of ester bond and the enzyme which catalyze the formation of this, that is called lipase. So, lipase enzyme help in the formation of ester bond with the help of fatty acid and glycerol. Now, see the structure of the phospholipids. We have seen the phospholipids are conjugated form of the fat which is made by fatty acid, alcohol and phospholipid group and the phosphate groups are there. Now, see which is in your syllabus, the structure of phospholipids. The phospholipids is also can you write easily, you can easily write the formula of glucose glycerol. We have seen in the triglyceride CH2OH, CHOH, CH2OH, here H is hidden, then this H and OH of the fatty acid, it is going to be combined, and for here you can easily write O C double bond O, that is R1. Same can repeat here O C double bond O R2. This is the same structure. Here the at third position there is a phosphate group, here O P OH, here O CH2 CH2, here is N, and this CH3 CH3. So, this is the structure of a phospholipid that is called lecithin, which help in the transport of molecules in plasma membrane. So, the phospholipid we can define as the phospholipids have only two fatty acid which are present at carbon 1 and carbon 2. They have phosphate group instead of third fatty acid. We have seen membrane like plasma membrane which surround the cell, and many structure within the cell are made by phospholipid bilayer. As the fluid mosaic model of the plasma membrane says, that is the trilaminar membrane where the bilayer of phospholipids are there. So, phospholipid is very important, which help in the formation of plasma membrane and many other structure inside the cell. Now, let us see the another structure of lipid, that is called steroids. Steroids, these are the lipids characterized by carbon skeleton. It consists of 4 fused ring. Example of steroid, that is cholesterol. It is also found in the cell membrane. Cholesterol is also the precursor of various hormone, some hormone here mentioned here, some hormones like estrogen, testosterone. Beside this, vitamin D, cortisone and many others. These are the precursors of these various type of important hormone, vitamin D and the cortisones. Now, see the structure of steroids. As we have already discussed, steroid a type of lipids. Here the common example of steroid, that is cholesterol. The cholesterol we have seen already discussed, it is made by 4 fused ring. So, make the 4 benzene like structure, this type. This structure is mentioned in your NCERT book. You can make easily the 2 benzene ring like structure, and continuously the other benzene ring like structure are this type here. this one is the third one. Now, the fourth one, this type. Now, here the second groups are attached. So, this is the structure of cholesterol. Here you find that there is the 4 fused ring 1, 2, 3, 4 and the other groups are also attached, that is called cholesterol. Here is 4 fused rings are there. It is very important biomolecules. It help in the formation of various types of hormones, just we have seen testosterone, estrogen, vitamin D and others. Now, nucleic acid. Nucleic acid, as you know, it is very important in the living system, that acts as a hereditary material that transfer the character from parent to the offspring. It helps in the formation of hereditary material. Now, as the term nucleic acid, this is acid like character which are present in the nucleus. We can define nucleic acid as it is a polymer of nucleotide. Polymer of nucleotides. These nucleotides are made by nitrogenous base, sugar and phosphate. Here we have already discussed in carbohydrate at the time of discussing pentose sugar. The pentose sugar is very important. There are 2 pentose sugar we have discussed. Name these 2 sugar pentose sugar, that is okay, very good, that is ribose and deoxyribose. This ribose and deoxyribose sugar are very important. It is the chief or main material of DNA and RNA. On that basis, the DNA or RNA has been named. Here the deoxyribose sugar are present, here ribose sugar are present. So, in case of nucleic acid which is made by the nucleotide, the sugar is ribose and deoxyribose, which is a pentose sugar, and the formula will be very good, C5H10O5. But in case of deoxyribose, one oxygen is less. In deoxyribose, the formula will be C5H10O4, because in deoxyribose there is the deletion of one oxygen at carbon atom 2 of the ribose sugar. So, on that basis the DNA and RNA has been named deoxyribonucleic acid, where the ribose sugar is in the form of deoxyribose and RNA ribonucleic acid, where the ribose sugar are present. So, firstly you can divide the nitrogenous base, and here the nitrogenous base are of 2 type. These nitrogenous base are made by 2 type of structure that has been divided. One is called purine, another is called pyrimidine. Purine we can easily understand it is two ring like structure. So, purine are made by two ring like structure and pyrimidine one ring. Purine, the example of purine, that is adenine, and second one guanine. Adenine can be abbreviated by A and guanine by Z. So, the purine is the chief constituent of the nucleotide that are, there are two examples of purine, that is adenine and guanine. Now, pyrimidine, it has single ring structure, that is thymine, abbreviated by T, cytosine, C, and uracil, that is U. So, nitrogenous base are of two types, purine and pyrimidine. Purine, adenine and guanine, pyrimidine, thymine, cytosine and uracil. Now, how we can write the structure of this purine and pyrimidine easily? So, here the some tips are there. The first of all, purines, which is double ring structure, the skeleton of purine, that is here nitrogens, and the another ring attached here, here is N, here is N. So, this is the skeleton of both the purine, that is adenine and guanine. Now, when we know the chemical name of the purine, adenine and guanine, we can easily write with the help of this skeleton, and by knowing the valency of carbon, that is 4, and nitrogen, that is 3. So, by knowing this, we can easily write the structure of adenine, all 5 types of nitrogenous base. Let us write the structure of adenine. First of all, you know that the adenine is also known as 6-aminopurine. First of all, name this. Here this one nitrogen is abbreviated as 1, 2, 3, 4, 5, 6. Here 7, 8 and 9. So, in purine 9 atoms are there, these are arranged in this fashion. So, if the adenine is known as 6-aminopurine, so at 6th position you place the amino group. Now, you can with the help of valency, you can easily write the structure of adenine. Now, here carbon has 1, 2, 3. So, one valency is left. So, here is double bond. Nitrogen 3, carbon here 1, 2, 3. So, it is not complete. So, in that cases here is the double bond. Here carbon 1, 2, 3, nitrogen is also not. So, here next double bond are present in between that. Now, here nitrogen has only 2 bond. So, here you place the double bond. So, with the help of bonding structure, you can write the easily the structure of adenine, that is called 6 amino purine. Now, see the structure of another nitrogenous base, that is purine, like guanine. Guanine, which is designated by G, its chemical name is 2 amino 6 oxy purine. So, now if we know the chemical name of the guanine, that is 6 amino 6 2 amino 6 oxy purine, we can easily write the structure of the guanine. Here is nitrogen. So, nitrogen has 3 valency, so it is also completed. So, all the valency of carbon and nitrogen is going to be completed. So, this structure is right. This is the structure of guanine. Guanine, the chemical name is 2 amino 6 oxy purine. Now, when we write the structure of guanine and adenine, that is purine, now you can easily write the structure of all 3 pyrimidine, that is pyrimidine. As we know, the pyrimidine is formed by the single ring structure. That single ring structure, when we know the structure of their skeleton, then we can easily write the all 3 form. The skeleton of pyrimidine, that is same as purine, but here difference is in purine the double ring structures are there, but here single ring structure are there. The first ring structure of the adenine and guanine right here, here carbon, here is nitrogen, carbon no doubt, here is carbon very good, here is carbon, and this carbon carbon is attached with the nitrogen. Okay, this is the skeleton of the pyrimidine. Now, first of all you see the structure of uracil. When we know the chemical name of uracil, that is 2,4 oxy pyrimidine. 2,4 oxy pyrimidine. So, name it, name slightly different from the purine. Here nitrogen is placed 1, 2, 3, 4, 5, 6. By knowing this, the placement of nitrogen and carbon 1, 2, 3, 4, 5, 6 position, you can easily write the structure of uracil. Uracil has 2, 4 oxy, means at 2 position and 4 position there is the oxy group. So, you can easily write here the oxy group with the help of double bond, here is the oxy group, double bonds are there. Now, you can easily write 1, 2, 3, 4, here the valency of carbon is satisfied, here 1, 2, 3 nitrogen has 3 valency. So, we can easily place hydrogen at nitrogen third. So, nitrogen here carbon at second carbon 1, 2, 3, 4, here nitrogen has only 1, 2, 3. We do not place the double bond here, because when we placing the double bond here carbon, carbon has 5 bond. So, nitrogen has attached with the hydrogen, here hydrogen is attached same as the purine. Now, in these cases the hydrogen carbon has 1, 2, 3. So, here is the double bond. So, we can easily write the structure of uracil with the help of the skeleton, and by knowing the chemical name of the all the pyrimidine, that is the uracil, it is designated by U. Now, same you can write the structure of cytosine here. Cytosine, first of all, name the chemical name of cytosine is 4-amino 2-oxy pyrimidine. By knowing this chemical name of cytosine and the skeleton of pyrimidine, you can easily write the structure of cytosine. Now, see here the skeleton is this, here nitrogen is placed. Now, 4-amino, name it 1, 2, 3, 4, 5, 6. Now, 4-amino at 4th position there is the amino group right here. At 2 position there is oxy group. By knowing this, here in all pyrimidine hydrogen is placed here. So, by knowing this structure, skeleton and chemical name, you can easily write the structure of cytosine. Now, see the nitrogen has 4 valency, no doubt. Here carbon has 1, 2, 3. So, and nitrogen has 2. So, we can place here double bond. So, here nitrogen has satisfied by the 3 valency, carbon has 4 valency. Now, here carbon 1, 2, 3, 4, no doubt here, but nitrogen has only 3, only 2. So, hydrogen is placed here. Now, in this cases carbon has 1, 2, 3. So, here hydrogen carbon has 1, 2, 3. So, double bond placed here. So, by placing this we can easily write the structure of uracil and structure of cytosine. Okay. Now, the last one pyrimidine structure, that is thymine. Thymine, its abbreviation is T and the chemical name of thymine is 2,4-oxy 5-methyl pyrimidine. So, you can write the skeleton of pyrimidine, that is 6 ring, 6 atom ring structure, and C, here C, carbon is here, and these 2 carbon attached with the nitrogen. Now, the name abbreviation number it 1, 2, 3, 4, 5 and 6. Now, with the help of this chemical name we can easily write the structure of thymine. 2,4-oxy at 2 position oxy group is there, at 4 position oxy group is there, and at 5 position there is methyl group. So, by placing this we can easily write the structure of thymidine. Here thymine, here the carbon has 1, 2, 3, 4 valency. So, here carbon has 1, 2, 3. So, by placing here double bond, here is hydrogen is, here nitrogen has 1, 2. So, 1 hydrogen is placed here. So, nitrogen has 1, 2, 3, carbon 1, 2, 3, 4, here nitrogen has only 2. So, hydrogen is placed here. So, you will see all the carbon and the nitrogen is satisfied with their bond. All carbon have 4 bond and nitrogen have 3 bond and oxygen have 2 bond. So, this is the structure of thymine, that is correct, that is thymine. So, by knowing this, we see the structure, we are able to write the all the structure of purine and pyrimidine, that is purine are of 2 type and pyrimidine are of 3 type, and by knowing this structure we can easily write the structure of the all 3 nitrogenous base. Now, the phosphate group, this is the nitrogenous base because nucleotide is made by nitrogenous base, sugar and phosphate. We have seen the structure all the 5 type of nitrogenous base, that is adenine, guanine, thymine, cytosine, uracil, which has been placed in adenine and guanine are placed in purine, thymine, cytosine and uracil are placed in pyrimidine. Now, the phosphate group is the chief constituent of the nucleotide. It is known as phosphoric acid. That is phosphoric acid. This phosphoric acid, that is H3PO4. This phosphoric acid, the phosphate and sugar which form the backbone of the DNA. The phosphate is H3PO4 in the form of phosphoric acid. This phosphoric acid have acid contain COH group and sugar contain OH group. This OH and COH group is going to be combined and it help in the formation of phosphodiester bond. At the time of formation of phosphodiester bond in the DNA, we will see later on. Now, this is the 2 things, that is the nitrogenous base and phosphate. Now, third one thing is sugar. We have already seen the structure of sugar. Sugar is pentose, that is C5H10O5. It has been divided into 2 group, all group contain aldehyde, that is ribose and deoxyribose. We have already discussed the structure of ribose and deoxyribose. Have a look here. The ribose sugar have skeleton this type, here is OH, H, H, OH, H, OH, H, CH2OH. By naming this 1, 2, 3, 4 and 5. So, this is the structure of ribose. At second position, at the time of writing deoxyribose, we can easily write the structure of deoxyribose. Here OH, H, at second position one oxygen is deleted. So, here is instead of H OH, here is only H, here H OH, here is H, here is CH2OH. So, this is the structure of deoxyribose and the formula will be C5H10O4. Here is the formula will be C5H10 H10O5. Now, after writing all these structure, now we are able to form the structure of DNA with the help of these biomolecules, and there is the formation of various type of bond, that is called phosphodiester bond and hydrogen bond and glycosidic linkage. The nucleic acid, the particles in nucleus of the cell responsible for the heredity called the chromosome, which are made up of protein and another biomolecules of the nucleic acid. Two type of nucleic acid have already discussed, DNA and RNA. Now, you see the structure of DNA are of this type, which is made by all three groups. Here is the 3 prime, 5 prime, here is the 3 prime, 5 prime. This is just like a ladder. The main bamboo of ladder is this, which is made by sugar plus phosphate. In DNA, that is deoxyribose and phosphate. In between that, adenine always pair with the thymine. One purine is always pair with the another pyrimidine. Cytosine always pair with the guanine. The adenine pair with the thymine with the help of double bond. Cytosine pair with the guanine with the help of triple bond. The diameter of DNA, that is 20 angstrom. So, the radius will be 10 angstrom. In one complete turn, the distance is 34 angstrom, and in this turn 10 nitrogenous base pair are there. So, each nitrogenous base pair have equidistantly arranged, that is 3.4 angstrom. So, this is the structure of DNA. You will see this structure in class. Now, think over this question, what are polyunsaturates? A lipid molecules contain dash molecules of fatty acids. My third question is, a purine has amine group on 6th carbon position is called dash. So, by knowing the all these purines chemical name, which I have already discussed, you may write this answer easily. Now, my fourth question is, a nucleotide consists of dash, dash and dash. Now, my last question, the glycosidic bond in DNA and RNA connect dash and dash. The glycosidic bond are found in between OH and OH group. So, you may find where the OH group are present, and this OH group liberate water and form the glycosidic bond. It help in the formation of DNA and RNA structure. Okay. With this, I hope you will easily understand the structure of all the lipids, nucleotides and everything related with the lipid and nucleic acid. Thank you.